All right, so we're going to start chapter 3. This chapter will be broken down into 5 different lectures so that they're a little bit more manageable. It's a long chapter, so make sure that you spend some time in each one of these videos and reading through the chapters related to that, to each video. And yeah, we're going to get started. So as soon as my lecture comes up, there we go. All right, so chapter 3, cells and tissues. This is part A. We're going to talk about the cell, just general functions of the cell, general makeup of a cell, where this first lecture is just kind of going over the components of what a typical cell would look like in the body. When we get to the next parts of the chapter, we'll go into specific types of cells and their functions. So this is just sort of background information and things you need to learn to understand what we'll talk about later with regard to tissues and cells, cells that make up tissues. So the cell we mentioned in chapter 1 is the smallest living unit. It's made up of organelles, and the cell theory states that cells are basically the building block of all plants and animals. So you are made up of tons and tons and tons of cells. Those cells group together to form tissues, and we'll get into that later. All new cells come from the division of a pre-existing cell. So you were once one cell from mom and one cell from dad that joined together to create one fertilized cell. The smallest unit of life that performs any physiological function is a cell. So if you think about a tissue or an organ that's made up of tissues, tissues are made up of cells, like connective tissue or muscle tissue. That's made up of cells that are performing the job of that tissue. So ultimately, you can be broken down into a number of different types of cells that perform a specific function, and that function helps keep you alive. This figure here, I'm not going to spend any time on because it just kind of overviews in a figure the cell theory. Occasionally, I will ask you to look at a figure while I'm discussing some other slides, so you can pull up the lecture slides or the lecture PDFs in Canvas and follow along with me. I will lecture about a slide that has text on it, and I will ask you to pull up a figure. So we'll do that here shortly, and you'll see what I mean. Cell differentiation. The word differentiation just means a cell is able to change and become something that's either more or less functional than the cell it once was. So there's a lot of different types of cells. They vary in size, shape, and function in your body. Those cells, but you ultimately came from one. And all cells in the body came from a fertilized ovum or egg from mom. That ovum that has been fertilized by a sperm divides into multiple cells. As those cells divide and create new cells called daughter cells, they specialize and change. That specialization and changing is called differentiation. So those cells that once came from a fertilized ovum differentiate, they change and specialize into 4 types of cells that are a part of 4 types of tissue. So if you are made of thousands and thousands and thousands of cells, each cell that has a specialized function is part of a tissue, and you have one of 4 types of tissues in your body. Epithelial tissue, epi, so this epithelial tissue, break it down. Epi means on top of. So epithelial tissue or above, epithelial tissue lines, like you can find epithelial tissue in your skin. So you can look at your skin, like look at the back of your hand right now. You're looking at epithelial tissue. It's on top of you. Connective tissue connects things and supports things. Muscle tissue and neural tissue. And each one of those tissues is made up of a particular type of cell, like an epithelial cell. We'll talk about the specific types of epithelial cells because there's not just one type. Connective tissue is made up of a number of different types of cells. Muscle tissue is usually made up of one type of cell, like a skeletal muscle cell or a heart muscle cell. Remember cardiomyocytes that we talked about back in chapter 1? Cardiomyocytes are a heart muscle cell. So that type of muscle tissue is made up of one type of cell. And neural tissue. The major cell type of neural tissue is a neuron. Again, a figure that illustrates that differentiation from an ovum that has been fertilized, here's a sperm, into one dividing cell that differ, and those cells that divide differentiate into these 4 different types of cells that make up these 4 different types of tissues. In the body, with regard to cells, there is extracellular. So extra is outside of or beyond. So outside the cell, there is fluid. That's fluid that basically fills the spaces between tissue cells. It's called interstitial fluid. That fluid is extracellular fluid that basically fills space, and cells reside in that fluid and float around in that fluid. The fluid inside the cell, so intra is within, intracellular fluid is the fluid inside cells. So your body is made up of just a ton of fluid. There's fluid inside cells, and it's trapped inside cells because cells have a cell membrane. And extracellular fluid is outside that cell membrane. So the intracellular fluid is also called cytosol. And sometimes it has a specific name like cytoplasm or cytosol. In muscle cells, it's called sarcoplasm. So there's cytosol is a generic term for intracellular fluid. The cell membrane separates those 2 fluids. So basically the cell membrane is a barrier that keeps certain things inside the cell and other things outside the cell, particularly fluid. And there's a figure that illustrates that. So this here is the cell membrane or plasma membrane. And outside that cell would be extracellular or interstitial fluid. And inside the cell is cytoplasm or intracellular fluid. This plasma membrane that separates the extracellular fluid from the intracellular fluid is created from a particular type of tissue or particular types of cells. Within the cell, basically each cell is surrounded by this plasma membrane. It's this barrier that separates the inside from the outside of the cell. The cytoplasm or intracellular fluid can basically be found within that cell membrane, encased in that cell membrane. And the cell membrane and the nuclear membrane, so the nucleus is an organelle, an example of an organelle inside the cell. That nuclear membrane is what surrounds the nucleus. And the cytoplasm or intracellular fluid varies with regard to its consistency within the cell. Organelles are found inside this, floating around in this fluid, intracellular fluid, and they are considered an organelle if they have a membrane. Typically, membranes are made up of what's called phospholipids. So membrane-bound organelles have a phospholipid membrane. The nucleus is an example of a membrane-bound organelle. There's also some that are not completely enclosed in membranes, and those are considered non-membranous, without a membrane, right? So this figure, keep it close by as we're talking about the next few slides, because you may be asked to identify particular components of this cell. So this figure shows you one whole cell and structures in and within and on this cell. So kind of the cell membrane here, this plasma membrane is here. There's actually structures that are created from the cell membrane. And then all of these intracellular structures within the cell here, we're going to talk about in the coming slides. And we'll overview them first and then go into some details about particular organelles. So follow me with this, stay on this figure, and then you can maybe pull up a double screen and keep this figure on one side from your notes, and then stick with the slides I'm going to be talking about next to identify those structures. And I will kind of flip around a little bit and refer back to that figure to point some things out. So have that figure nearby along with the next couple slides. So structures like peroxisomes, they contain enzymes that degrade within the cell. And they can break down fatty acids from fats or organic compounds. We'll maybe talk a little bit about these later. And they can neutralize toxic compounds. So there's 2 structures, 2 organelles within a cell that could potentially break things down or destroy certain compounds, substances that need not be in the cell any longer. Another example would be a lysosome. They contain digestive enzymes and not digestive like digesting food, but like digesting like breaking down. So to lyse, Lyse, something is to break it down. So lysosomes are these organelles that have enzymes that can basically break down components or compounds within the cell. And they can break down larger organic compounds or a damaged organelle that may need to be replaced, or pathogens. Some of the cells in your body, a lot of the cells in your body can engulf a pathogen, and by way of this organelle called a lysosome can break down that pathogen. We may talk a little bit about that when we get to the lymphatic system. And microvilli are extensions of the cell membrane. So basically, microvilli are folds in the cell membrane. So take a look back at that figure, or hopefully you have it by. Here's a peroxisome and a lysosome. Little tiny circular organelles that have a really big job to keep, basically the job of those two structures is to keep the intracellular environment safe, and will break anything down that doesn't need to be there or that could otherwise cause problems. Microvilli, follow my cursor, are here. So the cell membrane is this kind of pinkish, peach-colored structure. The cell membrane, it folds on itself, and those folds are called microvilli. And those microvilli are extensions of the cell membrane to increase the surface area of the cell membrane. And that by increasing the surface area of this membrane, you can increase or basically maximize absorption of extracellular materials. So the cell membrane isn't static. It can exchange or move things across it. So the more surface area that is available to do that, the greater the ability of that cell to exchange those materials. So it's a really, really efficient way. Like there's some cells in the digestive system that line the digestive system organs, and they're able to absorb nutrients that have been broken down or released from food. The greater the surface area, the more nutrients you can absorb. So it maximizes efficiency in those cells of absorption. The endoplasmic reticulum is a network of membranous sheets and channels. So I'm going to flip back to that figure and show you the endoplasmic reticulum. It's this blue structure here. See how it's just kind of these sheets or folds of an organelle? And there's two different types, smooth and rough. We'll talk about what causes the difference in those two. So here, this is the endoplasmic reticulum. And that endoplasmic reticulum produces, it synthesizes and transports substances. So we'll talk later about what type of substances, but basically anything that is created or synthesized in the cell body, in the cell itself, is synthesized and created in the endoplasmic reticulum. Works closely with the nucleus, so we'll talk about that as well. The smooth endoplasmic reticulum, or ER, has no ribosomes. And ribosomes, I'll flip back again, are those little darker blue dots. So think about them, they look like sprinkles on an ice cream cone or something. Those ribosomes, the smooth endoplasmic reticulum, we'll get to ribosomes next, synthesizes lipids, so fats, and carbohydrates. So there could be like a hormone, that's a steroid hormone, would be synthesized in the smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes, these little circular structures that are stuck to the folds of the endoplasmic reticulum, bound to the membranes of the ER. And they modify and package proteins. Proteins are synthesized in the nucleus and sent to the rough ER to modify and package those proteins. So the site of protein synthesis is often listed as the rough endoplasmic reticulum. So we're going to overview some of these structures, look at them, and then go into more detail about them in the next couple of slides. Ribosomes are these, they basically where proteins are synthesized. So they're bound to the rough endoplasmic reticulum. It's what makes them rough, makes the endoplasmic reticulum rough, the presence of ribosomes. Or they can scatter in the cytoplasm. And the job of a ribosome is to make proteins. So whereas the smooth endoplasmic reticulum synthesizes lipids, as we mentioned here, lipids and carbohydrates, the rough endoplasmic reticulum produces proteins. And you're basically made up of 3 macromolecules: carbohydrates, fats, and proteins. So ribosomes on the rough ER and the smooth ER are responsible for creating those functional molecules. The mitochondrion has a double membrane, has an inner membrane and an outer membrane, and this is the site of most of the cell's energy production, or ATP. So I'm going to flip back again, and you can see there's a bunch of them. It's actually pointed to here, but there's a mitochondrion, there, and there. So there's not just one mitochondrion in every cell. Some cells can have tons of mitochondria, and some cells can have very few mitochondria. It depends on the function and the necessity for ATP production. So some cells rely on tons of ATP production, others don't. And they can function a little bit better without a lot of ATP production. So the mitochondrion, it's nicknamed the powerhouse of the cell because it produces energy. Energy is ATP, and ATP is basically necessary for every cell in your body to function. Cytoskeleton is basically proteins that are organized into longer filaments or microtubules. Think about like a framework in your home. So like from what your home was built upon, there's likely a framework of wood, and all of those wood beams support the rest of your home. The cytoskeleton basically does that for a cell. It's able to move cell structures and materials around. So there's some proteins that will attach to cytoskeleton, either microfilaments or microtubules, and can move things throughout the cell or transport them. And it's also just a form of support, like the framework of your home. The Golgi apparatus basically takes proteins that have been created or synthesized in the rough endoplasmic reticulum and can modify them, change them to make them more or less functional, and then package them into vesicles. And a vesicle is another small membrane-bound chamber basically inside the cell that can be released from the cell. That Golgi apparatus, and then the nucleus is basically contains its own intracellular fluid, for lack of a better word. It's called nucleoplasm. It's fluid within the nucleus, and the nucleus is a membrane-bound organelle where proteins and DNA is basically located to help initiate the synthesis of proteins. It's surrounded by an envelope, which is basically a membrane, and that controls metabolism and protein synthesis. So DNA is converted to RNA, and RNA is then converted to a fully functioning protein or amino acid. So the initiation of protein synthesis happens in the nucleus. And those structures, I'm going to flip back again. Right here is the big nucleus. And then inside there is a nucleoplasm. Right here's the Golgi apparatus. So these little rounded structures are basically the vesicles that are holding proteins, making their way towards the cell membrane to be released. Nucleus. And the membrane itself is what's called selectively permeable. It has this property that allows certain things to move through and others not, typically based on size or charge. So the plasma membrane is a barrier. It separates the inside from the outside of the cell. And it's made up of a phospholipid bilayer. So there's two layers, bilayer, right? So there's a bilayer made up of phospholipids that have a head that's hydrophilic. Hydro is water, and philic is to love. So hydrophilic, the heads of the phospholipids, there's two components of one phospholipid. A head that likes water, so it kind of sticks out towards the extracellular fluid or in towards the intracellular fluid, and a hydrophobic tail. So a phobia is to be scared of something or not love something. So these hydrophobic tails are fats, basically lipids that don't like water. So the hydrophobic heads on one side of this bilayer, set two layers, points towards the extracellular fluid, and the hydrophilic heads on the other side of the layer point towards the intracellular fluid. And then the hydrophobic tails kind of touch one another. My PowerPoint's frozen. Hang tight, guys. There it is. Okay. All right. The cytoskeleton, so we're going to go from that kind of basic overview of what we talked about here and then go into some more detailed structures. Like the cytoskeleton provides an internal framework. It gives the cytoplasm strength and flexibility, and again can transport certain things throughout the cell. So there's a bunch of examples of cytoskeleton. So microfilaments, some locations and functions providing strength. Intermediate filaments, these are specific examples of cytoskeleton, types of cytoskeleton. Filaments here in skeletal muscle and cardiac muscle. The thick filament is made up of a particular type of contractile protein. There's where you can find it in the cytoplasm and then what it does, its function. Centrioles are really important. We'll talk about later in cell division. And then cilia are little projections off of the cell surface, the cell membrane. So they're actually embedded in the cell membrane, and they rhythmically beat to move fluids or secretions across the cell surface. So these are examples, specific examples of cytoskeleton. And there's some figures showing them as well, or a figure showing the specific examples. The rough and smooth endoplasmic reticulum. We've introduced this structure, but we're going to talk in some more detail about them. The rough endoplasmic reticulum has ribosomes attached to it, those little kind of circular structures that make it look, the appearance of the endoplasmic reticulum look rough. That's basically modifies and packages proteins for export to the Golgi apparatus. So the endoplasmic reticulum and the Golgi apparatus work really closely together, where proteins are being produced in the rough endoplasmic reticulum. They get sent, transported, or exported to the Golgi apparatus, and that's where those proteins that were produced in the RER get packaged into a vesicle. And a vesicle is bound, it's basically a membrane, a little teeny tiny circular structure that has a membrane that surrounds it. And inside that membrane, in the intramembranous spaces, are proteins that will be basically sent out of the cell or exported from the cell. The smooth endoplasmic reticulum lacks ribosomes. And that's where synthesis of phospholipids, cholesterol, which is a steroid, steroids. Cholesterol is basically the precursor for all steroids or steroid hormones. Triglycerides are an example of a lipid, and glycogen. Glycogen is a storage molecule for glucose, which is basically blood sugar. So the smooth endoplasmic reticulum synthesizes glycogen, and that glycogen is stored in the cell so that glucose can be readily available and broken down when it's needed to produce energy. So you basically take blood sugar, this glucose molecule, and convert it to a bunch of other molecules, substances, and in the process you produce ATP. So where mitochondria are producing ATP, they're using what was once glucose to produce that ATP. It gets converted to a bunch of things that you may have talked about in another class. But to store glucose as glycogen just makes glucose readily available. So the smooth endoplasmic reticulum has a pretty big job. That storage molecule is really important. And ribosomes can be fixed and stuck to or attached to the rough endoplasmic reticulum, or they can be free and scattered throughout the cytoplasm. And basically they're little manufacturing sites for proteins. And those proteins get sent to the Golgi apparatus. So you can see here, there's a blown up version of the figure, that cell figure. There's the endoplasmic reticulum, and you've got those little circles, darker circles are ribosomes. These would be fixed. Out here would be a free ribosome. This is what it would look like in a really large view. And then the smooth endoplasmic reticulum has no ribosomes. So you should be able to identify these structures in the cell on a figure and be able to discuss the function of these organelles. The mitochondria is so super important. The organelle is basically responsible for energy production, ATP production, meaning, by way of aerobic metabolism. And aerobic means with oxygen. So oxygen is used inside a mitochondria to help in the production of energy. And that's called aerobic metabolism. You also have what's called anaerobic. An is without, or A is without. So anaerobic metabolism occurs in the cytosol or cytoplasm without the use of oxygen. So you can still produce some ATP, not a lot, by way of anaerobic metabolism. Aerobic metabolism occurs within the mitochondria, and tons of ATP is produced. And like I mentioned earlier, the number of mitochondria per cell varies depending on the needs of the cell. It has two membranes, an outer membrane that surrounds the organelle, basically creates the organelle, the mitochondrion itself, and an inner membrane that folds into what's called cristae. And that increases the surface area that's exposed to the matrix, which is the liquid basically enclosed by the inner membrane. It's called the matrix of the mitochondria. And that's where metabolic enzymes within that matrix basically produce ATP. So here's a mitochondria. There's the outer membrane and the inner membrane. And that inner membrane then folds. And those folds are called cristae. Those folds increase the surface area to maximize the movement of everything needed and production of enzymes that are needed to produce ATP. So it's really important. This Golgi apparatus, from where the endoplasmic reticulum produced proteins, the Golgi apparatus is made of flattened discs called cisternae, and they receive transport vesicles from the rough endoplasmic reticulum. So the Golgi apparatus then takes those transport vesicles, and a vesicle is just bound by a membrane, and does a couple of things. It allows basically the plasma membrane properties to change the sensitivity and the functions of a cell. So the Golgi apparatus has a membrane renewal vesicle. So it can produce a vesicle that has a membrane around it. So it's creating this little kind of chamber. A vesicle is a chamber that has a membrane around it. And that membrane can basically fuse with the cell membrane of the entire cell and renew parts of the cell membrane that maybe are old or need to be replaced. It also produces what's called a secretory vesicle. And that secretory vesicle has within it something like a hormone or an enzyme that was produced inside the cell and needs to be released from the cell. So basically these secretory vesicles can travel from the Golgi apparatus where they're produced, holding and containing hormones or enzymes, and then those hormones or enzymes get released from that vesicle as they pass through the cell membrane. Lysosomes are another type of structure that are produced in the Golgi apparatus. And lysosomes, as we mentioned earlier, contain these enzymes that the inside of the cell can use to break down substances or molecules that are not needed, or no longer needed, or would otherwise cause problems. So here's the example of the three different functions of the Golgi apparatus. So here are the folds called cisternae. And as those folds produce whatever they need to produce, like a part of the membrane that needs to be renewed, basically that just pinches off. There's the membrane here, and that vesicle travels to the cell membrane and fuses its membrane with the cell membrane and replaces part of the membrane that needs to be replaced. That same thing happens when a secretory vesicle is produced, but within that vesicle, can you see the little tiny dots in there? That vesicle makes its way to the cell membrane, fuses its membrane with the cell membrane, this red here, and in doing that, it exocytoses or releases the chemicals that were inside, or the molecules that were inside that vesicle, like a hormone or an enzyme. The third would be the lysosome. So it produces this membrane-bound structure, and within there's enzymes that can break down components of the inside of the cell that are no longer needed. Those lysosomes have three basic functions. They can fuse with another organelle and digest it or break it down. They can fuse with a vesicle from the outside of the cell and digest it. So there may have been a vesicle that made its way in, and that vesicle that made its way into the cell needs to be broken down. or it can release the enzymes into the cytoplasm that can result in maybe in a cell that's injured or is dying. It can release enzymes that would degrade the cell itself. And in degrading the cell itself, it causes what's called autolysis, or the cell basically destroys itself. So here there's the figure of the processes. You can follow along or rewind part of the video and look at this figure and then listen, or take a look at this figure and then rewind part of the video where I was talking and kind of follow along those steps. The nucleus is typically the most prominent and visible organelle because it's pretty big within the cell. It's the control center, basically. It's the brain of the cell. It has a nuclear envelope that surrounds it and basically creates the nucleus. And there's pores, which are passageways that allow chemical communication between the nucleus and the cytoplasm. So lots of materials can be exchanged back and forth through these pores. Like ions and molecules that would regulate, that can be regulated by proteins. There's the nucleus, the nucleoplasm inside there, and there's nuclear pores that we talked about. The nucleoplasm is basically the fluid component of the nucleus, and it contains filaments that are there for structural support, but also ions, enzymes, a little bit of RNA, and DNA. So the DNA is encased in your nucleus. And your nucleolus is a nuclear organelle. So it's actually an organelle within the nucleus. The nucleus is an organelle within the cell. And that's where the nucleolus synthesizes ribosomal RNA, and that also assembles ribosomes. You can find them most prominently in liver, nerve, and muscle cells. So not every nucleus has a nucleolus, but most liver, nerve, and muscle cells have that. DNA within a nucleus basically stores the instructions for protein synthesis. So DNA has the building blocks within strands that coil around themselves. Basically, there's a double helix. Looks like a slinky and another slinky kind of came together and smashed into one, for lack of a better explanation. In non-dividing cells, DNA is loosely coiled that forms thin filaments called chromatin. We'll talk about that. So here's chromatin right there. All of those strands are all kind of winding pieces of DNA. Right here would be DNA itself, a double-stranded helix. So again, kind of looks like a slinky pulled apart and then wound around another one. That double helix, basically at the beginning of cell division, the DNA coils, the tightened coils become more complex and they form distinct structures called chromosomes. Think about it, you have chromosomes, you have 23 pairs of chromosomes. And basically at the beginning of cell division, you make a copy of those chromosomes that can be held together at what's called a centromere. So I'll show you that on the next slide, and you can flip forward to the next slide if you're looking at the notes right now. Each of those duplicated chromosomes is called a chromatid, and you've got 2 chromatids for 1 chromosome once it's been duplicated. And basically, somatic cells, which are every cell in your body, each cell has 23 pairs of chromosomes. So here's the chromosome. When a cell begins cell division, those supercoiled DNA molecules become chromosomes. So this is a chromosome. And then before the cell divides, so that in cell division, the 2 cells that result from cell division each have the number of chromosomes necessary, 23. So this would be 1 chromosome. And then that chromosome basically duplicates itself, and but it stays connected at what's called a centromere. Think the center of this chromosome is a centromere. So this is 1 chromosome, and this is still 1 chromosome. It just has twice as much DNA, so it's duplicated itself. Each one of these duplicated chromosomes is called a chromatid. The structure of DNA has these long chains, basically held together by hydrogen bonds. And DNA is made up of what's called nucleotides. Nucleotides have a base. It's called a nitrogenous base. And there's one of 4 bases: adenine, thymine, guanine, and cytosine. So these are one of 4 bases that a nucleotide can have. And that basically stores information in what we call the genetic code. A sequence of 3 of these nitrogenous bases is called a triplet, and that codes for a specific amino acid. That triplet is also called a codon, C-O-D-O-N. If you've heard that word before, it's also called a triplet. And that's what codes for an amino acid. And amino acids are the building blocks of proteins. So this coding sequence is what tells the cell, or tells the nucleus, other components of the nucleus, that those 3 nitrogenous bases, or those 3 nucleotides, will code for a particular amino acid. And this basically is the functional unit of a gene. And that DNA, basically all the DNA in your cell, in your nucleus, basically contains what's needed to produce a specific protein. And proteins are fully functional molecules that perform jobs of the cell or jobs in the body. So here's a strand of DNA that's just been lengthened and kind of unwound. And you can see C for cytosine, T for thymine, there's A for adenine, G for guanine. Each one of these 1, 2, 3 triplets is a codon, and that codon basically says, okay, these 3 nucleotides, or these 3 nitrogenous bases, are what make one particular type of amino acid. And they can be grouped into different triplets to create a different amino acid. You have a bunch of different types of amino acids in the body that create proteins. And protein synthesis starts with DNA. So these strands of DNA separate. They get basically detached from one another, disassembled. And an enzyme basically assembles then nucleotides into a single strand of what's called messenger RNA, or mRNA, by basically complementing the base pair from the DNA. So there's pairs of bases, nitrogenous bases, that will kind of connect or pair with one another in a DNA sequence. Like adenine pairs with something called uracil. In RNA, uracil is the replacement for thymine, that U here. And guanine pairs with cytosine. So those 2 pairs are the only pairs that can occur when creating mRNA. So if a U is available, or if an A is available, it'll pair with a U. Or if a T is available, it'll pair with an A. So in the DNA sequence, thymine is the nitrogenous base. In an mRNA sequence, it's uracil. So the pairing works in the opposite direction. But if there's a T available in the DNA, it'll pair with an A, an adenine, on the mRNA sequence. But if there's an A adenine base in the DNA sequence, it'll pair with a U, a uracil, in the mRNA sequence. So you're basically creating a brand new strand of messenger RNA by complementing or creating new pairs with the DNA bases. And 3 RNA nucleotides is a codon. And that codon is what codes for an amino acid in the protein. So DNA is converted to mRNA, and mRNA is converted to a protein. And that protein is basically created by codons, a sequence of 3 RNA nucleotides at a time, that can then, this mRNA passes through the nuclear pore in the cytoplasm, and it heads to where in the cell? The endoplasmic reticulum. So here's what it looks like. You've got a DNA strand that gets disassembled. And basically that disassembled DNA strand has these base pairs, A, T, C, and G. Those base pairs are then used to produce mRNA, this green strand. And that mRNA, messenger RNA, is produced that is complementary to the DNA strand. So again, if there was an adenine here, the mRNA strand would contain a thymine. Or if there was a cytosine here, the mRNA strand would have on it a guanine. So you create that mRNA strand, and then it gets shuttled to the endoplasmic reticulum. Transcription takes place in the nucleus, and transcription is the process of copying DNA and to create mRNA. Translation occurs in the cytoplasm on ribosomes that are composed of proteins and ribosomal RNA. So transfer RNA, tRNA, carry amino acids to the ribosomes. So they transfer one amino acid that's made up of a codon, 3 mRNA bases, nucleotides, to ribosomes. And basically that nucleotide sequence is called an anticodon on each tRNA. So there's a tRNA that has an anticodon that's basically complementary or the opposite of what's on the mRNA sequence. And that anticodon binds to the matching codon on mRNA and then starts to result in this chain of amino acids that string together to form a protein. So here it is in the figure form. 1, transcription occurs in the DNA. DNA is copied to mRNA. So you're creating this mRNA strand that gets sent out of a nuclear pore and into the cytosol. So this pink or peach area is the cytosol. Ribosomes, basically, translation is where a translation occurs. And translation is the process of converting an mRNA sequence to a functioning protein. Translation occurs in the cytoplasm on ribosomes composed of proteins and ribosomal RNA. So here we are. The complementary binding occurs between anticodons of transfer RNA, tRNA, right? So there's some amino acids, right? So there's mRNA, this green strand, and transfer RNA are these little kind of bluish purple structures that transfer amino acids, right? So they transfer a codon, which is basically 3 nucleotides of the mRNA strand. So basically pick off a codon at a time, 3 nucleotides from the mRNA strand, and transport them. And they transport them to build a protein. The ribosomes, basically, the chain of amino acids is built into a protein. So using the mRNA strand, amino acids are assembled, basically, to create a protein. So this little transfer RNA, right, there's little codons or amino acids that all string together to form a protein. And those ribosomes detach then, leaving the protein molecule behind the mRNA and the ribosome. So all of this is one full process to create a protein from where it started in the nucleus as DNA. mRNA is transcribed and then sent out of the nucleus through a pore. And then ribosomes and tRNA, transfer RNA, basically take that mRNA strand, which is more of a code, and then create amino acids. And those amino acids then string together to form a protein. When cells are getting ready to divide, so all that occurs, protein synthesis, at fertilization. Basically, you're one single cell. And when you are mature, you're roughly 75 trillion cells. So that one cell eventually divides and divides and divides into what you are as 75 trillion cells. Cell division occurs in 2 ways. One way is called mitosis, which is basically how most cells divide. The somatic cells divide in that manner, that all have 23 pairs of chromosomes, so 46 chromosomes complete. And then meiosis occurs to produce sex cells in the human with only 23 chromosomes. So basically when mitosis occurs, mitosis is cell division in most cells in the body other than sex cells. So the egg in the female or ovum and the sperm in the male. So mitosis is when any cell divides, it divides into 2 identical cells. So when one cell has 46 chromosomes, it divides, it duplicates its DNA, as I mentioned earlier, and then divides into two brand new cells that both of which have 46 chromosomes. In meiosis, one cell that has 46 chromosomes divides and produces daughter cells with only 23 chromosomes, so that daughter cell, either the ovum or the sperm, can, upon fertilization, create one fully functioning cell with 46 chromosomes. The lifespan of a cell basically varies from hours to decades. It depends on the cell. Some cells self-destruct after a period of time. It's called apoptosis. And some cells can live for a really, really long time. This cell division, so here's the normal original cell. And then the division of two cells, this is mitosis. These daughter cells are identical to the parent cell, the original cell. Most cells spend most of their time in what's called in a phase of the cell cycle called interphase, where the cell is fully functioning, it's performing normal functions, and no division, it's not undergoing division. When the cell prepares to divide, chromosomes duplicate themselves and basically create what we mentioned earlier was a chromatid, and then proteins start to get synthesized that will help in cell division. Mitosis creates identical copies of those chromosomes, right? So during interphase, the latest stage of interphase, just before mitosis occurs. So there's three phases in the cell cycle, three major phases in the cell cycle. We'll break these down a little bit further. Interphase is where cells spend most of their time. And then division is mitosis. And cytokinesis is basically the very end of once mitosis has occurred, the cell officially divides. It separates into two daughter cells. So we'll talk about that. There's a figure that describes that. Again, cells spend most of their time, my cursor there, in interphase, and then spend a part of their time, their living time, in mitosis. And the very end of mitosis is cytokinesis, or the actual division of cells into two. So when the DNA in interphase, when the DNA replicates itself, it basically duplicates the amount of DNA. So all 46 chromosomes double the amount of DNA that they have and create a sister chromatid using a DNA polymerase. A polymerase is an enzyme that binds to exposed bases on DNA and basically promotes the binding between bases that are complementary, and it links them by covalent bonds, by way of covalent bonds. So this DNA polymerase basically unwinds, or some enzymes that unwind DNA, the DNA polymerase helps to duplicate that DNA. So think poly means multiple. DNA polymerases basically duplicate that DNA in each chromosome. So there's a newly formed DNA strand. So the DNA gets unwound by enzymes, and then new DNA strands, double helixes, get formed here to double the amount of DNA in each chromosome. So you don't increase the number of chromosomes. You always stay with 46 chromosomes. You just double how much DNA is located in each chromosome. So that when you split the cells, you can send the exact amount of DNA that was in the original cell to the new cell, both new cells. And at the end of DNA replication, the original strands of DNA have completely separated, and there's two brand new identical strands of DNA molecules. So you've got twice as much DNA in the chromosome as you had before replication started. Centrioles are the center of that chromosome. So think about the arms of the chromosome and other enzymes that have proteins that have been synthesized. So as the cell leaves interphase and proceeds and heads into mitosis, you have twice as much DNA as what you started with. It's doubled. And then once you head into mitosis, here's where we begin cell division. Prophase is the first phase, where centrioles start to move to the opposite side of the nucleus. The centrioles are these structures that chromosomes can attach, will follow. There's little spindle fibers that connect a centriole pair. During prophase, these spindle fibers and centrioles start to move to opposite sides of the nucleus. And then the nuclear envelope, the membrane in the nucleus, starts to disintegrate. And the chromosomes tightly coil together so that you can actually see them under a microscope. When prophase is complete, metaphase occurs, and the chromatids align at the center. It's called the metaphase plate, the center of the cell. The fourth phase, and I'll show you this on a figure, and we'll go over it again, third phase is anaphase. So as centromeres, not centrioles, of each chromatid pair split. So the centromere is basically what holds a chromatid to another chromatid. And two chromatids are part of a chromosome held together at this centromere. So if you're not sure what I'm talking about right now, go back and look at that figure that I showed you earlier of a chromosome, where there was one chromosome, and then it duplicated its DNA into two chromatids, bounded at the center by a centromere. During anaphase, each of those chromatids that have the identical DNA split and move towards either side of the cell, where centrioles are located. Telophase is the last phase, where the nuclear envelope reforms. So the nucleus becomes a membrane-bound organelle again and gets larger, and chromosomes uncoil to chromatin, and the cytoplasm basically constricts and forms a cleavage furrow. So the cell membrane starts to pinch in on itself so that division can occur, which is called cytokinesis. So right here is interphase. Normal cell, you'll see a nucleus that you can identify under the microscope. Well, that nuclear membrane starts to dissolve in the first phase of mitosis, which is prophase. So there's where the nucleus once was, and there's the chromosomes that are kind of increasing in their density, and they're showing up, whereas they weren't showing up here in interphase. You can now see chromosomes, and these two little dots here are centrioles that are starting to move towards either side of the cell, and that'll help pull the chromatids in either direction during anaphase. So here's an example, two examples of prophase. Right here is this chromosome. So this whole structure is a chromosome. Right there's the centromere. Each one of these arms of the chromosome is a chromatid, and it is identical to the other. So that in metaphase, these chromatids line up at the metaphase plate here. So there's a chromatid and a chromosome and a chromosome. There's an example of it under the microscope. And then as anaphase begins, the chromatids split. So right here's the centromere. This chromatid will go to one side of the cell, and this chromatid will go to the other side of the cell. So right here it's happening. There's one set of chromatids headed in this direction towards the kind of upper left, and another set of chromatids headed towards the lower right. So then during cytokinesis, the chromosomes that are here get stuck in this cell, and the chromosomes that are here get stuck in this cell. And that's right, that little pinching in here is a cleavage furrow. So here's one brand new cell and another brand new cell. Cytokinesis is the division of cytoplasm. So brand new daughter cells are created, and each one of these daughter cells is identical with regard to its DNA to the original cell from which it came. And that's where we're going to finish this lecture. This is the first of 5 lectures that you'll see in chapter 3. I'm going to continue with the next, and go ahead and click on that, pull up the new PDF, and we'll go from there.